Mitochondrial DNA is one of the main causes of genetic diseases in today's society. It is caused when one or more mutant mitochondrial DNA is paired with a normal mitochondrial DNA, causing heteroplasmy. In 2009, the Oregon National Primate Research Center, or ONPRC, began researching and experimenting with rhesus macaque monkeys in order to determine whether or not they can eliminate this disease. During reproduction, the mitochondrial DNA is inherited from the maternal parent's oocytes. In order to counteract this, the ONPRC hypothesized that by taking a karyoplast that had a spindle chromosome complex in metaphase stage 2 and placing it within the donor oocyte's prevetaline space, they can eliminate the mutant DNA from the patient. This is the prevetaline space seen here, and once the karyoplast has been placed within the space, it then needs to initiate fusion between the karyoplast and the cytoplasm. Two methods of this was tested to see which was safest and most successful. The first was electroporation and was successful within an hour. However, the pulsation from the electroporation caused spontaneous maturity from metaphase 2 through to anaphase. The second method was introducing the spindle chromosome complex to an extract of the Sendai virus before placing it in the prevetaline space. This was highly successful within half an hour and was able to postpone maturity until intracytoplasmic sperm injection could occur. Both methods were tested in fertilization. Electroporation continued increased prematurity and went through to the 8-cell blastocyst stage, which was further than was needed. This made it undesirable in this experiment. Sendai virus was much more successful and continued standard rates of fertilization, cleavage, and blastocyst. It can be seen here that the Sendai virus is much more compatible with the control than the electroporation was, which makes the Sendai virus the more successful of the two and will be used for the remainder of this experiment. The successful oocytes were then fertilized in vitro and placed within a selection of rhesus macaque monkeys for the remainder of the term. This was needed to decide whether or not the gene replacement had been successful. Seen here is the stages in which the embryos had been placed within their recipients. On the 24th of April 2009, the first set of twins was produced from mitochondrial gene replacement and was followed closely by a third on the 8th of May. Once they had been deemed healthy, they were then tested to see if any mutant DNA had been carried over. By looking here, you can see that all infants were homoplasmic, and the only mitochondrial DNA present was healthy mitochondria from the egg donor. This result suggests successful transplantation without malformed mitochondrial DNA. From this, we can conclude that through this series of experiments, basic mitochondrial gene replacement in rhesus macaque monkeys is a successful method for mitochondrial gene replacement. Thank you.